All right, hi, welcome to my kitchen. I'm about to break my fast and eat breakfast, and normally I'd be going off to school after this to teach about the digestive system. But because of COVID-19, we are staying at home. It is really important to stop the spread of the virus. So I'm going to bring you some lessons straight from my kitchen. In our class right now, we're trying to build a human body. And so it's a really complicated task. So it got me thinking about where should we even start? And when you think about it, our digestive system is just one big long tube. And when the cell is first forming, it forms something called a blastula, which is a group of cells. As that blastula forms from the zygote, the one fertilized cell, it starts to make a tube. And so that tube goes right from our mouth to you know where. So I'm cooking up my breakfast and I want it to be so good because I am what I eat. You are what you eat. We are all doing this process of alchemy. Do you know what alchemy is? Alchemy back in the day was when scientists and alchemists, they were called, tried to convert base metals into gold. And we do kind of a similar process. We break down food and turn it into energy. We break down food and turn it back into the things that we need to help our body grow. Okay, so as I'm making my oatmeal, I'm thinking about trying to make it really balanced. So I have my oats and I put in there some walnuts, which are a source of fat. My oats are a source of carbohydrates. And I cook it all up with some milk, which is a source of protein. And sometimes I get fancy and put collagen in there, which helps to strengthen fingernails and hair and joints. The collagen helps build cartilage. I also really like to put apples in my oatmeal, but some of these apples are so big I can't possibly eat a whole apple at the same time eating my oatmeal. So you notice what happens as they start to brown. So that got me thinking, I wonder how we can get these not to brown. I want them not to brown. So I've tried a bunch of different things. I seal them up in Tupperware. But then I was thinking way back to when my mom and my sisters used to make apple pies. And my mom, if we made too many of them, she would say to us, oh, put some lemon juice on them. And so I got wondering, like, what does lemon juice do to stop browning of apples? Do you have any idea? So today we're going to try to figure that out. We're also going to figure out how to run a scientific experiment in your kitchen. We're going to figure out how our food even gets digested. So if I eat this breakfast now, about 4 hours later, where will it be? 3 days later? How much food do I need to eat and what do I need to eat to build a healthy body? So that's what we're going to go over today. I'm really excited because in a couple weeks we're going to hear from a different perspective. We're going to go through today from the scientific perspective of Western science, and then we're going to hear from a Ayurvedic practitioner who's going to teach us about the digestive system. Okay, so let's start at the top, shall we? Let's set up an experiment to try to figure out how this apple turns so brown and what we can do to solve it. Okay, so my first step is going to try a couple different solutions. So I have here my vessel. I'm going to go and try lemon juice, vinegar, a control, and water. And in order to set this up, I want to cut the surface area of my apples about equal. Okay, so you can't really see this, but I'm just going to, let's see here. Oh yeah, let me set this up. Okay, so here we are. I have my apple, I have my 4 slices of apple, and I'm going to, here's my experimental design. And as I'm going through this, I want to make sure that every step along the way is consistent for every single slice of apple. So one of these apples is going to be with lemon juice. So I'm going to dunk it in the lemon juice, have it in there for 30 seconds, take it out, and put it in my lemon juice compartment, all nice and labeled. Then I'm going to take the next one, and I'm going to do vinegar. Do you have any idea what I do vinegar? So the same reason why lemon juice is sour is because it has a low pH. And things that have a low pH are called being acidic. So other things that are acidic in your kitchen are things like vinegar. Okay, so I'm gonna put some vinegar in there. Then I'm going to take, one is going to be just dunked in water. Can you think of why I just dunk one in water? And one, I'm not gonna do anything to. This serves as my control. This is the part of my experiment where I get to measure what happens when I don't do any treatment to the variable. So I don't add any treatment. This is the only one. I get to compare then my results to this one and say, this one browned a lot and the other ones, I'm gonna wait 30 minutes and we'll come back and see what happens to our apples. All right, so what's in the food that we eat? This apple, for example, is made up of a sugar called fructose. Can you think of other things that are made up of fructose? That's right, fructose is a fruit sugar. So a lot of different fruits are made up of fructose. There's another type of sugar called glucose, and that is what we refer to as our blood sugar. Ultimately, when we start the process of digestion, we go through a big long tube, lots of series of tubes, and the whole process is to break down our big components, our carbohydrates, our proteins, our lipids, and our nucleic acids. We want to break those all down to their component parts, and then our bodies can either use those for energy or use those to build our bodies. So this apple is made up of fructose, and there's actually a couple different types of sugars. So what I'm going to do is I'm going to just go ahead and start the process of digestion, and I'll show you something you can mess around with at home. And remember, the difference between science and messing around is writing it down. So go ahead, and I encourage you to take notes on what you do in the lab. And this is scientific data that you're collecting to support your own conclusions. It can happen right here in your kitchen. Okay, so starting the process of digestion, I take a bite. This is mechanical. I'm breaking down the apple to have a larger surface area, right? From that one big bite, I'm gonna end up with a lot of pieces. It's gonna be kind of gross right now, but digestion is pretty gross, so. All right, I have formed what is called a bolus. That is a bolus. It's a ball that's been partially digested by mechanical forces and also chemical forces. So in your saliva, there's water and mucus and these things called enzymes. There's one particular enzyme that works in your mouth called amylase. And amylase works to start breaking down starches, which are complex sugars. Complex sugars are present in one of my other favorite foods, cookies. In this cookie, I have a polysaccharide called starch. And as I start to break down the starch with the sugars in my mouth, with the enzymes in my mouth, I break it down into a simpler sugar. Well, here we go. Creation of another bolus on the way. And you know that the amylase is working. Try this. Eat a piece of bread and notice what happens. Oh gosh, spit. Oh, I'm gonna try that again. You shouldn't talk with food in your mouth, right? But I'm gonna do it. As you chew, things get sweeter because you're breaking down this big molecule into a smaller one. Alright, bolus on the way. Gross. Alright. So for this next part of kitchen chemistry, I want you to rifle through your first aid kit or your backpacking gear and try to find some iodine. Iodine is used to sterilize wounds. It also can be used to sterilize water when you're backpacking. So you might have eaten some iodized water. It works from preventing you from getting things like giardia. But it doesn't taste that good. Okay, so I'm gonna mix my first experiment here. I have fructose from my apple. And I have a molecules. I'm gonna make a solution. So you can work in your kitchen with making lots of solutions, different solutions, do this every day. But a solution is one thing dissolved in another. So for example, you might make a saltwater solution, which is what we're gonna do later today when we go look at our brine shrimp. Check out that experiment going down. Okay, so here we go, our two different vessels, and we have our iodine. Iodine is an indicator for starch. So what do you think is going to happen? Well, I'll just show you the control. This is what iodine looks like regularly. This is brown, orange color. And notice no change because there's no starch. Now I'm gonna put it in a vessel that has starch and start to see what happens. You're observing a chemical change. Under the presence of a starch, iodine will turn purple. So now you can start to design an experiment around this question. How does my spit, my saliva, start to work to break down food? What is the effect of saliva on food? And you can use iodine as your indicator solution. You can also just go around and test which items in your kitchen contain starch. All right, another fun thing you can do with iodine, which really gets at the key point of the digestive system, which is surface area. Our digestive system would be nothing without the millions of folds and increased surface area that happens from our mouth all the way down to our small intestine and our large intestine. So here's the next experiment. Potatoes, which are also have a lot of starch in them. Okay, so with our potatoes, we can add iodine to 3 different size cubes. What you're gonna find, you can do some simple math to figure out the surface area to volume ratio. So cubes that are larger have a lower surface area to volume ratio. For their volume, they have less surface area. But smaller cubes have a larger surface area to volume ratio. And that means that more of the iodine can get in more of the potato. This is really important when we think about why cells are so small. They need to be able to have as much surface area as possible. Cells are pretty cool too because they can change their structures to accomplish the function. So if what they need to do is to absorb more nutrients, they can create extensions, things called villi in your small intestine or rugae in your stomach. And those extensions serve to increase the surface area from the normal circle-sized cell. Okay. All right, so now we're gonna get to see visually the movement of the iodine into our potatoes. And remember that the iodine will start to turn our potatoes purple. You can see that starting right there because they have starch in them. So we're gonna wait for 30 minutes and cut open each one of these different size cubes and see what happens. All right, so we just went over 2 types of carbohydrates, or our sugars. That supplies most of the body's energy. There's another type, so there's fructose, fruit sugar, glucose, blood sugar, starch, which is a complex sugar, and then we have what makes up plants, and that's called cellulose. So here's an interesting question for you. How is a broccoli the same or different than a Brussels sprout? Do you know? They're actually from the same family called the Brassica family. These are the flowering heads, and then this is the bud. This is a plant that has been genetically modified to make a bunch of different types of vegetables that we like to eat, from cauliflower, Brussels sprouts, cabbage, kohlrabi, kale. They're all versions of the same plant, the Brassica. It's the Brassica family tree. Okay, and they all come from one plant called the wild mustard plant, okay, which came from Central Asia. We can do that better. Okay, what else is in the food that we eat? So in my oatmeal, I had oats that provided the sugars, the carbohydrates, the complex starches, and apples that provided fructose, the fruit sugar. Those are all going to get broken down into one form of sugar called glucose. There's another polysaccharide or complex sugar that is present in a lot of the foods we eat, things that are green. And it's also known as fiber, but the technical name of it in biology is cellulose. So in our broccoli, we find a lot of cellulose. This makes up the structures and the nice woody, hardy part of vegetables. Here's another interesting little tidbit for you. How are broccoli and Brussels sprouts similar or different? They're actually both from the same family, the Brassica family. The broccoli is the flowering part, and the Brussels sprout is the bud. Scientists have used selective breeding to bring up traits that they really like. So if you really like Brussels sprouts, you might also really like cabbage. This is the larger selectively bred version. So what they did is they took a Brussels sprout that was really big, a plant that produced really big Brussels sprouts, really big buds, and they mated it with another plant that produced really big buds. And over time, they selected the ones with the biggest buds, and they got a different version called cabbage. Other members in the Brassica family include kale, kohlrabi, spring greens, and cauliflower. They are a really good source of phytonutrients, which are nutrients that your body needs, like vitamin A, D, E, K, vitamin C. There's two different groups of these vitamins. They are fat soluble and non-fat soluble. So we haven't even really gotten to our fats yet. So in my oatmeal, what was the source of fat? I had some walnuts and I cooked it in milk. So milk contains fat molecules, and you know that because you might go to the store and get 2% or 4% or whole milk. That is the amount of fat that they have suspended in the solution. Walnuts are a good source of fat. They supply omega-3 fatty acids, along with salmon and tofu. Avocados and eggs give us omega-6s, which are a type of fatty acid that we can't actually make in our body. So it's important to get those in your diet. What are fats? We have two different types of fats. We have saturated fats and unsaturated fats. And the difference is, we need to use our bodies and do some kinesthetic movement here to figure this out. If you are a lipid, you're a fat, you have a tail here. That's this fatty acid chain. Carbons, hydrogen, oxygens make a chain. If you are a saturated fat, that means all of the tails are straight and you can line up next to each other. That means they're solid at room temperature. But imagine, so that's a saturated fat, you have an unsaturated fat, you have a kink in the carbon chain. So if I make a kink in the chain, I have a double bonded carbon. That gives me an unsaturated fat. Now it's a solid at room temperature. So things like this, this is actually clarified butter, this ghee, which I made, which is a really fun recipe if you want to be at home making some really healthy, very nourishing item. And yeah, coconut oil, also a saturated fat, solid at room temperature. Unsaturated fats, I might look up here. Here I have oil. I have all different types of oils, right? Vegetable oil. We're gonna run a little experiment with vegetable oil. What happens when you mix it with water? I'm sure you might know the answer here. Let's see here, I'm gonna use this. You might use this at home to do an experiment and try different types of oils. Try making a lava lamp, that's pretty fun. Okay, so if I take my oil, I can see what's happening. They don't mix, right? And the oil is actually forming what's called a micelle. So all of the tails, the fatty acid tails, they don't like water. Obesity, they're hydrophobic. And so they hide from water and they form their own droplets. And that's the oil droplets here. Lipids are really key when we think about how things get in and out of our cells. So every one of our cells is made up of a phospholipid bilayer. So there's 2 layers of lipids. And it's a phospholipid, so it's a special type of lipid. And it actually has a head of it, which is a phosphate group. And the head likes water. It doesn't mind water. It's called hydrophilic. And the tail, remember, just like our oil, doesn't like water. So the tails are going to hide from the water and the heads are going to go towards the water. And do you remember what the watery substance in the cells is called? That's the cytoplasm. So as you have one layer lines up like that, and then on your other side you have another layer and another head. And so this forms a barrier between the outside and the inside. And along the length of the bilayer you have pores. And the pores allow materials in and out. It's kind of like this door. If I'm a cell and I want to let something in, like some glucose, I open up my door. And if I don't, I close it. I like to then imagine all of our cells are kind of like a plastic bag. They have that membrane, phospholipid bilayer, separates the outside from the inside. And it's something called semi-permeable. So things like water can go in, but bigger molecules need to be let in by the door. All right, so what happens if I add soap to my mixture? Soap is a really cool molecule called a surfactant. And what it's able to do is grab onto both the hydrophobic part of the oil, and it also has a hydrophilic part, which is the water. So when I wash my hands, you notice how all the molecules are a little bit more integrated right now? I wash my hands and the soap molecules grab onto the grease and the dirt and the grime, and then I put them under running water, and all of that flows away. The virus that we're fighting right now, it has a membrane coat, and remember how our membranes are made up of oils. So when you wash your hands, the soap does its work to get rid and dissolve the membrane of the virus, and then the virus can't live anymore. Although, is it actually living? Because it just kind of parasitizes. It is a parasite to our cells. It only has DNA and just the membrane, no other organelles, so scientists say it's not living. That's kind of an interesting discussion. So that brings us to another experiment. Is all the food we eat alive? So we know that Brussels sprouts, the cabbage, these are all part of living things. Living things provide us with vitamins and minerals. For example, this ginormous carrot Carotene helps our eyes function well, so we notice that people who don't eat a lot of carrots and don't get enough carotene had bad eyesight. This is how we determined a lot of which vitamins that are essential for humans to use. Certain vitamins are either fat soluble or not fat soluble, so A, D, E, and K, those are all our fat soluble vitamins. And vitamin A and C are water soluble, meaning they turn into a solution with either fat or water, right? Because either certain things will dissolve in fat and certain things will dissolve in water. They're very different. So we might ask, they were living at one point, so does everything we eat have DNA? You can actually do this experiment yourself at home, and I would love if you do. The directions are fairly simple. I'm not going to do it for you today. It makes a little bit of a mess, but it works under these principles. You need to dissolve the membrane of your cell. So you're going to add soapy water. You're going to make a solution of water and soap. And you're going to add it to your cells. Your cells come from any living thing you want to test, anything that you think might have DNA. And I'll give you a little hint. Strawberries work really well because in each one of their cells, they have 8 copies of DNA. In each one of our cells, we have 2 copies of DNA. So once you dissolve the membrane, you release the DNA. Then you can add a little bit of salt, which actually helps break apart these proteins that hold the DNA together. And so when we talk about epigenetics, that is this idea that your DNA is modified based on your environment. And that kind of gets back to whether or not they have proteins that sort of lock them up. They're called histones, and the histones can be have acetyl groups or methyl groups. Anyway, so the salt breaks apart the proteins that hold. It's kind of like, imagine a big long chain of yarn, and you roll it all up in like on a carton or something, and it helps keep it together. Okay, so you add salt, and then you take it, mash it all up, put it in a bag, put it in a blender. It releases your DNA. Throw it in cold isopropyl alcohol at 70%. You can find that in your first aid kit as well. When you take that, mix those two together, the DNA is not soluble in the isopropyl alcohol, and it will precipitate or come out of solution. Then you should be able to find there's a thin line there, and you can pull out the DNA. So if you end up running that experiment, let me know. Test different types of food and see if it has DNA in it. You don't need to eat meat to get protein. What you can do is you can eat things like lentils. Which brings us to another little growing experiment. If you want to see, you can sprout your own lentils. Maybe you want to see the difference between red lentils and green lentils. That's kind of interesting. Or you want to watch them grow. You put them in water, make sure when they're germinating, when they're sprouting, they actually don't want to be by light, so you cover them up first. Okay, so this is a fun experiment you can do. And again, the difference between science and messing around is writing it down, so make sure you write it down in your science notebook. All right, let's go ahead and wrap this up. Let's go over what happens in our digestive system. So I've eaten my delicious oatmeal, which is now cold, but it's so good. It's so good. I'm chewing it. I'm creating a bolus. And then the process of swallowing is actually really neat. You have a coordinated effort between a flap above your pharynx, which is your throat, which wants to cover your larynx, your windpipe, and then your epiglottis, which opens up to allow food in to your esophagus, which is a big, long 10-inch tube, which is super muscular, and it squeezes down the length of it and circularly, two different motions of squeezing, has striated muscle tissue, and it plops that food down right into your stomach. At your stomach, you have all these different rugae, increased surface area, and what I'm gonna do right now is I'm gonna show you some pictures from a really neat book. All right, so I'm gonna use this incredible book, The Incredible Machine by National Geographic, to guide us through what happens when food enters your mouth and goes all the way down this big, long, 3-storied tubes, 3 stories of tubes, all the way down to your anus. All right, so let's start here. Someone tell me what this is. I think this is incredible. This is a scanning electron micrograph of, guess what it is? After you eat, you might go brush your teeth. That's what this is. This is a toothbrush bristle on your teeth. And that there is some plaque. Plaque buildup from bacteria that like to feed on what's left behind in your mouth. All right, and here's our tongue. Your tongue is a powerful muscle capable of many feats. And below here, these are actually nodules, lymphatic tissue, that help guard against infection. There's your tongue. Your tongue takes the bolus and pushes it back so it can hit your esophagus. As it travels down your esophagus, there's 2 different motions, and they're called peristalsis. And the peristalsis moves the food from your throat all the way to your stomach. It gets plopped down here in the stomach. That's the image of it there. And here you can see all the hills and valleys of the stomach's inner lining. And this actually flattens out when it gets food in it. Food's gonna stay here for about 4 hours. Selectively, some of it gets to leave and some of it stays. Once it hits the stomach, it gets these messenger signals hit a cell, and they are able to tell it, release the gastric juices, which are made up of pepsin and hydrochloric acid, a scorched earth approach to just getting all of your food to be broken down to its component parts. All right, and then it gets let in selectively by the sphincters to the small intestine. And your pancreas, which is right behind your stomach, actually releases a alkaline solution. So when we're talking pH, we have basic solutions, which are 7 and above, and acidic solutions, which are 7 and below, 7 being neutral. This, our pancreas. We have our stomach acid that comes in, all those gastric juices. And I'm going to add an alkaline solution. What happens, we are very aware of, is an acid-base chemical reaction, which produces bubbles here and also neutralizes. This is then going to become a mixture with a pH of 7. Here's a picture of the small intestine, magnification 300 times. All right, and this is the increased surface area we're talking about. Those are the villi. You can see here, this is a cross-section of a cell, and that is the structural adaptation. That's the villi that come out. And this is actually a glob of mucus, which helps to move the food along. In the small intestine, there's some more mucus. There's actually some even more mucus. Mucus, very important here in the body. After the carbohydrates and proteins are broken down in the small intestine, it goes to the large intestine, which is large because of its diameter. It's actually 5 feet in compared to the small intestine, which is 15 to 20 feet. It completes its journey. So we have reabsorption of the water, and finally, what's left, the nutrients, are absorbed into the bloodstream. That gets sent to our liver, which is right here. The liver is a 3-pound organ, and it serves over 500 functions. Some of those being breaking down fat, carbs, into blood sugar. It detoxifies the blood. It distributes, it stores and distributes nutrients. All right, here is a cross-section of the liver, which I think is a really cool diagram. You can see that the cells are lining inside this incredible maze here. And this is the blood vessels that go through carrying the blood. We have red blood cells, white blood cells. And this is how it gets filtered. Actually, also bile comes out, breaks down fat. Bile is kind of like detergent, helps to break down fat. This is an experiment they ran. They tested a kid. They tested kids to see how they responded to sugar. And then they did IQ tests on them, and they found that kids who ate high processed sugar had lower IQs. Kind of interesting, something to think about. But you also need to fuel your brain. You need to make sure you eat all the nutrients that you need in order to be able to get glucose to your brain to help it do its powerful actions. Just as a fire takes oxygen to burn, in each one of our cells, there is a fire burning, even without a flame. What happens is that the mighty mitochondria, food gets taken in, broken down, each one of the it's in a small form called glucose, and the carbon molecules on the glucose are blasted apart, releasing energy, or ATP. In the process that takes oxygen, there's a series of enzymes that help out. And that ATP is kind of like the currency of the cell. Imagine taking $1, going and taking it to the bank and getting 100 pennies. that is like breaking down glucose to have ATP. ATP then can be used to go do all your cellular functions and your big body functions like walking, breathing, and even smiling as your muscles start to move to make a smile. And that takes breaking an ATP molecule apart and releasing the energy. So you can think about that next time you put on a great big grin. Maybe you're doing that right now as you learn about the digestive system. If your fire goes out, that's a bad thing, right? So if you don't have enough glucose to go around, your liver, that incredible organ, actually does another task. What it does is it takes glucose and stores it in a molecule called glycogen. That is a storage molecule for the food that you just broke down, and it can be used when you're in between meals, when you're starting to feel like, oh, I'm kind of hungry, it's been 4 hours, my stomach's emptying, and you can rely on that glycogen. Okay, so we've covered a lot today. We've gone over a couple different science experiments. I will check up on some of these. We have why do things brown? We can kind of notice our control seems pretty brown compared to lemon juice, vinegar, and water. I'll cut these apart in a minute here and see how far the iodine is able to move in at 3 different sizes to look at our cells. Okay, so we have small, medium, large. Let's go ahead and cut them open and see how far the iodine moved in. So here's the largest one. See, it's just on the surface there. Let's see this next one. Not too much more either. Let's see the smaller one. Got a little, oh yeah, here you can see kind of how it started to happen. It has started to move down into our little model of a cell. So not too much moved in on the larger ones. Definitely is not reaching the center, but here this has a little bit more likelihood to. That's kind of the point there. And to wrap up the whole life cycle, these were once nice pears and they've been taken over by a fungi that started to digest them. And while we have that internal digestive system, fungi actually digest things extracellularly outside their own cells. So you can see this is the end of the road, and it will all get broken down by the fungi back into its component parts, back down into sugars, which are going to get eaten up by the fungi. Thanks for coming along on the journey with me. I hope you run some experiments in your own kitchen, and I can't wait to see what you post.